Welcome, everyone. Thank you for joining us for today's webinar, In Vivo Surgical Models for Drug Discovery, Hidden Pitfalls and Their Avoidance. It is presented by Matthew Flegal, Senior Associate Scientist in Behavioral Pharmacology and Surgery at Biogen. My name is Judy O'Rourke, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Matthew Flegal. I will now turn the presentation over to him.
Hello, very nice to virtually meet all of you, and today I am going to be covering several things related to how to properly use, I should say, in vivo surgical models for drug discovery. As the title suggests, there are plenty of traps and pitfalls to step into, so I'm going to try and give you kind of a jumping around overview of things that would be very useful for you to keep in mind when you're planning these types of studies. So to start off with, this is me many years and many pounds ago. I've been doing this for quite some time, and again, I have been able to see an awful lot of mistakes and errors, but also learn from a lot of very skilled and knowledgeable people in how to do these types of studies and have you get the data that you actually need. You need them to be successful. So again, it's just a little bit of overview. Some of the things that we're going to touch on are fairly advanced surgical procedures. And so I put in things like this is a aortic transplant that I had done several years back, Faraday aortas, CNS surgery in canines, and so forth. And the thing to keep in mind is when we think about the possibility for complications in vivo surgery, these are the kind of procedures that we're thinking. We're thinking of something that is comparatively rare, is comparatively seldomly done, and only a few people do. And if you get nothing else out of this talk, what I'd really like you to get is all of these things matter at the most basic surgical procedure that you could use in your discovery pipeline. It's not all transplants, central nervous system surgery, and what have you. Putting in a cannula improperly for drug delivery can completely destroy the results of your study. So the concepts that I'm going to give you, again, kind of a high-level overview on, they're applicable across the board. Please don't think that just because you're doing a commonly performed surgery, that the requirements of it are somehow less. Surgery is surgery. You're invading the body cavity, or invading the underlying tissue of a lab animal, a living creature. It's fraught with opportunities for mistake, and we're also going to need to talk about the welfare component of it, because I hope it goes without saying, if you're in the lab animal field and you're not focused on the welfare of the lab animals, I'm going to speak from my own biases and suggest you might want to find an alternative, because that's not a good place for you to be. We'll touch on that a little bit. All right, so here's the key, if you will. All of the molecules that you come up with, all of the devices that your engineers develop, at some point in the process, they're going to have to go into lab animal studies for efficacy, for safety testing, for toxicity levels, and there's probably going to be several of them in a fairly well-defined pathway. And unfortunately, Oftentimes, the people and the groups who are conducting these studies are kept very separate from the actual discovery teams. And that's unfortunate because the level of work and the applicability and translatability of the work that the in vivo study teams are doing is going to directly affect the success or failure of your compound or device. Put more simply, if studies run poorly, you can have a good compound fail, you can have a bad compound have its failures masked until much later in the discovery, development, even into the safety process, when you've spent hundreds to thousands of FTE hours, you've spent millions to tens of millions of dollars. And in this day and age, when we're all trying to make do with less, whether we frankly want to or not, we don't have the luxury of wasting these kinds of resources. And it should go without saying as well, you're not just wasting resources, you're wasting the lives of the lab animals that you're working with. And I would propose to you that if that is not a major concern to you, if the welfare of your test subjects is not a critical piece of what you're concerned about, you should probably seek, let's call it, another career path. So it's important. And to amplify that just a little bit more, they are a specialty in their own right. Please don't ever assume that an in vivo study is a basic thing that requires minimal skill and minimal experience. And I've heard numerous times over the past 25 years the horrible quote, well, anyone can probably do it. And that's not completely untrue. Anyone probably can do it. It's just they're going to do it really, really poorly. If you've invested a major portion of you and your team's life and budget into, again, a molecule or a therapeutic or a device, you kind of owe it to yourself to make sure that the people putting it in are doing the best job possible. And the thing with surgery is, 25 years ago, surgery was probably not as significant a component of most drug discovery pathways. But in this day and age, there aren't a lot of compounds that make it all the way through the discovery and safety process without going into lab animals that had some sort of surgical intervention. Could be a radiotelemetry implant for cardiovascular monitoring, could be a catheter for drug delivery or sample collection, but it's pretty safe to bet that at some point in the pathway, somewhere down the line, someone is going to be doing surgery. Boy, you better hope that they're doing it right. We'll talk about why. So things to think about when you're planning that study. And you as a discovery team should always have these thoughts. Because again, if you pick the wrong species, if you pick the wrong strain, your study may fail through no fault of what you're using, but because they selected the wrong animal. The anatomy is different. Keep in mind, most lab animals are quadrupeds. They walk around on all fours, which means that their organs are pushing on different tissues. Their anatomic structure is different. And if you think that doesn't affect how your molecules and devices work, I'm afraid to tell you, you're completely wrong. The effect of stress on the heart changes the heart position. You know that. Quadrupeds versus bipeds will change things. You need to be aware of that going in. Size. If picking something topical in the news, if you're looking at traumatic brain injury, with all of the things, either with NFL players or soldiers in combat, a 2-gram brain in a mouse cannot be accelerated to reproduce the damage of a 2-kilogram brain in an adult human. There may be other ways around it, but if you're trying to blindly mimic something, the anatomy itself, the size and weight of the animal, may make your results wrong, to be blunt. And there's anatomic variance even between the strains. One strain of mouse, C57 black, is not the same as Balanced. A Lewis rat is not the same as a Sprague-Dawley rat. A purpose-bred beagle dog is not the same as a hound. A cynomolgus non-human primate is not the same as a rhesus. There's anatomic variance between those traits. They will matter. Could be their brain anatomy, which may directly affect how your compound works. It could be the thickness of the skin over an implant. It could be something as somewhat esoteric, but when you're doing, for instance, ocular work, corneal toughness and the fibrotic structure within it completely change the reaction, not just to an implant, but an injected test artery. And also just keep in mind, these strains will drift over time. The C57 mouse that you got 5 years ago will probably have some genetic drift from one that you've done today. Talk about some of the implications of that as well. But here's one of my favorite examples. I teach a lot of brain surgery to a lot of people, and even down to mice. If you read the Stereotaxic Atlas, it says at the very beginning, first page, that all of these stereotaxic coordinates you're going to use for drug delivery or Alzheimer's or ALS or MS or what have you are based on only 26 animals, one strain, at one age range. They even specifically say mice vary much more in brain anatomy than rats, but rats do as well. But people tend to look at a reference, so they'll find a paper, talk about papers, that they will blindly follow without making sure that it works. So there is a real world effect of having these anatomic differences between animals. So I'm not going to spend a lot of time focusing on kind of the overview of selecting the right model. There are good texts out there. I would recommend reading them. But little things like, are you trying to model the cause of an injury or a surgical intervention or a dosing paradigm or the result? And neither one of those is necessarily all right or all wrong, but you need to have these types of questions answered up front so you can select that proper model. You have a question to answer. I would very strongly recommend, based on seeing some real successes and seeing some groups with real failures over the years, if you're executing a study based purely on someone said I had to do it, and I'm checking an arbitrary box, yes, I have done this study, but you haven't worked out why you're running that study, chances are you're going to design it poorly. And yes, you'll check that box, but you won't get the information that was the original reason that you were required to check that box in the first place. You can prove the wrong hypothesis. You can provide ambiguous results. And at some point, and again, this day and age, probably in a very constrained time frame, you're going to have to make a decision based on the results that you get. And to be blunt, if they're garbage, your decision is so much more difficult. And if you've erroneously set up a study to make your compound, again, fail, utterly fail, you're going to proceed with the wrong data. And I would propose to you, if you don't have this type of information, wait. Doing a bad study rarely, if ever, is an advantage over just not doing the study at all. It's better to have no data than compromised data, because you're still going to make a decision, but you won't know that it's compromised. And again, to circle back, if you do a bad study, those are animal lives that you're wasting. Again, that should be a pretty significant concern for you. So when you're looking at models, and just in general, what do? We go to ComMed, we go to PubMed, we go to the internet, and we start looking up journals and we find papers. Papers are wonderful as jumping off points, but especially when it comes to in vivo work, they tend to be far less useful than you might hope for. Simple things. Can you reproduce that study in-house? It is frightening how often you can't. And you're also, if you're not looking with a somewhat skeptical eye at the paper, are you sure that the author chose the right species, the right strain, the right technique, the right materials, the right approach? One of the ones that I've seen for years and years and years to this day, they're looking at the histopathology of a surgical site. They're looking at an implanted device. And they use highly reactive materials to just place the device. So if I close an incision with the most reactive suture material, as far as I know, in the entire field, silk, I've completely destroyed my histopathology. It's completely affected. Any effect that I might get from my implant or my biomaterial or my injection at that site is probably masked by the fact that I have a hyperacute inflammatory reaction just based on the suture I put in. And to this day, I see that in papers. So don't assume that they did things right. And there are red flags. So when you're looking at an in vivo study, look for things like odd group sizes. If they have 7 groups and group 1 has 8 animals, group 2 has 3 animals, group 4 has 5 animals, and group 5 has 1 animal, it may be because those animals are very difficult to get. They're a genetic cross or what have you. My experience has been when you see that sort of thing in a paper, it's probably because they had a very high mortality rate and they're only reporting the ones that survived, which might call into question the procedure that they've done. If they have 100% success, if everything looks so perfect that it looks like it was drawn, it may be. We're all susceptible at times to cherry picking data and what have you. And if the study looks too perfect, it may be. And it's worth hitting that with a skeptical eye and investigating a little bit more. You're also going to find there's often very minimal technical details. I mean, I have seen transplant papers that said, we transplanted 1 heart from a donor, and that's the entire sum of their technical procedure. There's a lot left out there. So last thing that I would do, I'll tell you what I typically do. And it's so much easier than when I started. Reach out to the authors of the paper. Talk to them directly. It is amazing the important nuggets of information that are left out of a paper, especially when it comes to technical details in surgery. And don't be surprised if when you reach out, you find that the person that you talk to who's actually doing the procedures is not reflected on the paper, and it is not uncommon for them to respond with something along the lines of, yeah, that's not how we actually do it. And then you can get the correct information, because I can tell you from bitter personal experience, there's nothing better than spending 3 months frantically and with great frustration trying to reproduce a paper to finally reach out to the surgeon in it and find out that there were 2 or 3 key pieces of information that hadn't been included. Better not to waste your time, and again, better not to waste the animal lives. Last, call it kind of an overview, is make sure you're gathering data. Lab animal is a fountain of information, and you should be collecting it all. If you're handing this off to an in vivo team, are they doing even a somewhat cursory full necropsy at the termination of a study? Just because you have a central nervous system drug doesn't mean that it's not having unexpected effects on other organ systems. And when you do a post-euthanasia necropsy and you find that the kidneys are 4 times larger than they should be, or their heart is dramatically distended in the atria, was that what you expected? No. Was it an organ system that you were looking at? Still no. Is it something that you probably want to know as part of the discovery team about probable complications? Absolutely. So you have to look for these things. Are the people trained properly? Do they have a protocol? Not just for necropsy, but are they watching the animals during the course of a study? Are they looking for untoward outcomes? Again, you're passing your molecule and your device off to a team who they can completely destroy your research. It's worth a little bit of due diligence to make sure that they're doing it properly. So here's one of my favorite examples, because it's one of the simplest things that we do in lab animal. It's dosing. We do oral gavage, which If we can do it, that's what we're going to want to do. It's fairly straightforward. Technically, it's ideal for translatability because most of your medicines, most of your molecules are going to be administered orally if at all possible. But there's a problem. How you do it can directly affect the outcome of your study. pH. If you have a neutral pH test material that cannot survive changes in pH and it's dependent upon absorption in the large intestine, and you put it into an acidic GI tract, your molecule is probably broken down before it's ever absorbed. It'll look like a failure. And it's technically not, because you never had the chance for the medication to actually get taken into the animal. Is the tube tip placed properly? Is it placed all the way in the stomach so that your entire dose is administered that way? Or is, possibly due to technical inexperience or what have you, is it placed in the esophagus as well, so the tube running from the mouth to the stomach, and half your test material is passed back up and spit out onto the cage floor? We also, little things like, do we use the full volume? So here's the maximum volume that you can give to an animal. If it's been eating and its stomach is full and you administer a max volume, well, now you've distended the stomach. And what's the normal GI reaction to that? It flushes out the GI tract as quickly as possible, typically liquid. So what's going to happen is the test material that you just administered, that you were expecting a certain degree of uptake over 12 hours of GI transit, it just got shot out the other end in half an hour, and none of it has been taken up. And that's going to be variable across your animals. So again, even something as straightforward as an oral dosing of a medication, if it's done improperly, can completely destroy your study. I'm not going to brush briefly over training, but the people doing your study and doing your surgeries, are they trained? Have you validated that they're doing it properly? And when was the last time you validated? Surgeons drift. We change our technique over time, either because we've learned things or we've modified our technique because it's easier, more straightforward, or what have you. But it can mean that we're not doing surgery the same way that we were when we validated on the model. And maybe that won't make a difference, but maybe it will, and it's important to know that. If you have multiple surgeons, have you validated across them? Are they all performing the surgery the same way? Again, the most basic surgical procedure in live animal medicine is probably placing a catheter for dosing. If I put the catheter in a different place than another surgeon, I may directly affect the uptake on the animal or create complications in that animal. The most basic surgery. But if there's variability between surgeries, the results are going to be different from surgeon to surgeon. And how do you analyze those results? Do the surgeons have good time management? Are they using proper ergonomics? Are they causing themselves fatigue over the course of their day so that the animals that they do at the end of a surgery day take twice as long and have significantly more complications? Are they managing themselves so that their quality of work stays consistent? We've seen for years and years, one of the truisms is mix up your test groups over the course of the day because you don't want to have this time effect. Once upon a time, we thought it was due more to the physiologic rhythm of the lab animal itself. Taking a cue from human medicine and surgery, we've kind of learned at this point it's because the surgeon fatigues and gets sloppy or is rushing or wants to get to lunch or wants to go home on time, and they compromise their work based on that. Again, it's a little thing, but it will affect your study. And then specifically in surgery, what's the health status of your lab animal? This is one of the biggest pitfalls I've seen. I've been a bit of an advocate for a good decade and a half about this. And is this: if your animal is not healthy, if it has compromised organ systems, this will change the results of your experiment. Full stop, no argument, it will change your results. Just looking to make sure that they're walking around and eating is not sufficient, especially if you're trying to affect a target organ. If you're doing brain surgery, you probably want to make sure that you're doing it on a healthy brain. If you're doing cardiovascular surgery, you probably want to make sure that you have a healthy cardiovascular system. And this became very clear to me about 17 years ago when I started doing cardiac ultrasound on every lab animal that I did surgery on. In large part at the time because I just learned how to do ultrasound and I needed as much practice as possible. But this is my test case. This happened 3 weeks into this process. We had a non-human primate that was going to radiotelemetry surgery, had a veterinary exam, perfect blood values, perfect veterinary exam, looked perfectly healthy. And when I did ultrasound, I found an intraventricular septum that was so large that it basically closed off the ventricles, which are the 2 chambers that pump blood either into the lung or throughout the body. The cardiac volume on this animal had been chopped over in half. The amount of blood that was coming out of the heart was barely a third of what it should have been. And this animal looked perfectly healthy. We analyzed the tissue. It had cardiac hypertrophy, it had degeneration. This animal would have gone on study. It would have given us data that we would have used to evaluate test materials, and they would have all been compromised based on this. And I'll tell you this, and I apologize, this is unpublished data, but for what it's worth, at this point, I have done cardiac ultrasound on approximately 2,000 rats. I'll use them as an example because it's my highest end. About 7% of them had significant cardiac deficiencies, typically ejection fraction, typically ventricular hypertrophy, but that's 1 out of 12 was significantly compromised. And if you think that won't matter to your study, unfortunately, you're wrong. So to focus a little bit more like a laser on surgery, aseptic technique is critical, absolutely critical. We're minimizing contamination of our surgical site. We don't want to create subclinical or clinical infections. Subclinical and clinical infections will change your results. Subclinical infections, so it's an infection that is systemic through the animal, but is not giving you grossly observable signs. So you don't see virulent discharge, you don't see lethargy, you don't see noticeable compromise, but their vasculature is carrying infectious microorganisms. Chances are they have partial renal failure. Chances are they have deficiencies in cardiac output in the heart. They have changes in brain chemistry. If you have a subclinical infection that, because of a breach in the dura, because you've done brain surgery, crosses into the brain, you have transient lesions which fibrose afterwards. This isn't a maybe. This is almost a certainty. Again, that is going to change your data. That is going to change your results. Aseptic technique is critical. We do it in humans. All of our lab animals that we typically do surgery on are mammals too, and they get infected just the same way. Again, we slow down their healing. This is based on toxins, the physical debris. We change the local circulation, increase the animal's pain and discomfort, which again is an animal welfare issue. But as I said, even if you can't see the infection, if it is present, it is dramatically changing all of the physiologic parameters of your animal. And again, your data has completely changed. And unfortunately, if you don't do blood work, if you're not conscientious in your technique, you may well be introducing these problems and you'll never know. You can go on for years without realizing it. And trust me, having worked with some groups who've had that horrible awakening, you don't want to be in that group when they realize it. And just a quick study that I'll throw out there for aseptic technique. This is going to hit specifically with biomaterial implant surgeries. I've taken this to the person who originally taught me surgery a quarter century ago. Dr. Vince Mendenhall did this type of study, and it's a bit eye-opening, which is simply take a surgical implant that is proven safe and non-reactive. You take 12 rats, and you dip your porous implant in Staph aureus, which is a common contaminant microorganism. You just wrap it around their finger. And then 28 days later, you sacrifice them. In the animal that everything was done properly with pure aseptic technique, you have great ingrowth into the fibers themselves. Everything looks wonderful. You have wonderful continuity for collagen fibers. Again, everything looks fantastic. You send it to your pathologist, this biomaterial, this implant that you've been working on for the past 5 years. They look at it and they say, it's great material. Continue development on it. Got something. Whereas much lower magnification, if you do it and it is contaminated, the bone dies. Because the product, you have edema, the implant is pushed away, you have a complete inflammatory cascade. You send it to the pathologist, they're going to tell you that your implant is failed. The material is too reactive, and it's not. It's based purely on what you did and how you did it. And yeah, they may be honest enough and direct enough to say maybe your surgery was compromised. But I can tell you, I'm aware of several biomaterials dating back to the 50s that we ran one study, because again, you usually only get one chance at this. It's done poorly, the pathology looked terrible, and it took decades for that material to be resurrected. Again, you've gone through so much work to develop your molecule, you've gone through so much work to develop your implant. Don't let it fail because of poor technique on the surgeon. So how do you get your surgeons to be doing it properly? And I throw out there a bit of a philosophy that I like very much, which is treat it as a discipline, treat it as an art. If you're in surgery, you should want to be in surgery. You should be passionate about what you're doing. Because if you think about it, how I do my techniques can completely change the results. Again, a plastic surgeon who gets rid of a scar, it has the exact same surgical toolbox as a surgeon who leaves a giant scar. Why do they have different results? What are their different outcomes? What are their different plans when they go in? And I put in here, if you're a surgeon, keep in mind what is important, what is not. I put this picture in as kind of a quick story. When I was just learning surgery, I was trying to make the prettiest incisions possible, doing knee replacements in beagle dogs. And I had the most beautiful closures possible. You couldn't see them. They were perfect. What I did is I sacrificed the strength of my closure for the appearance. And so after a few hours, when the animals, unfortunately, they were on opiates, so they had plenty of pain medication, they had tranquilizers. As soon as they started moving, the incision started to break down. So I spent about 7 hours in the evening until about 2 o'clock in the morning, re-anesthetizing and re-closing every animal and doing it properly. Because I went in there with the wrong goal. I was trying to make it pretty, but I needed to make it strong. So if you have surgeons and you want them to get to that point, here's what they need to do. And I'll warn you, especially in some regions of the country, this may be a little bit disturbing, so just please keep that in mind. But here's my philosophy in a nutshell. How did Tom Brady get to 6 Super Bowls? Like I said, this may be triggering for many people out there, but I'm from New England. Practice. That's what we're surgeons need to be doing. This is a skill set that takes a lot of work to get good at. Again, I'm not just talking transplants. I'm talking the most basic procedures that you do. It takes practice to get good at them. It It takes practice to maintain proper asepsis, minimize damage to the tissues, and maintain integrity of the organ systems. You have to practice. And I'm going to throw out there's a difference between practice and repetition. Practice means trying to do better. It doesn't mean just going through mechanical motions. I live in Massachusetts. I work in Cambridge. I spend almost 3 hours commuting every day. I can tell you right now, my commute does not make me a better driver. I'm not trying to learn to be a better driver, but I'm putting 3 hours a day and getting no better at these skills. So it's not just doing the procedure over and over again. It's trying to do better. It's looking at pathologies, investigating all of your results, looking for complications, trying to understand why you had them, and then fix them. Many ways you can do it. Set yourself little challenges. Can I work with an incision that is a centimeter smaller, which is better for the tissue integrity? Am I following my cases? Every animal that I do surgery on, I keep tabs on it for the life of that animal, because especially with device work, you can be without complications for months and then have a problem arise. And at that point, if you're not observing your animals, you'll never know. And I wanted to put this in here, but let's not forget, anesthesia is an art too. And I put this in, this was a summer project for one of my technicians, Teresa, who did fantastic work. And what she did is she simply went in mice and rats to see if we were seeing similar things that we had seen in large animals. So anesthesia, injectable anesthetics, a lot, especially with rodents. We've depressed their central nervous system. We've depressed their respiratory centers. We've reduced their inspiratory and expiratory volume in their lungs. Without supplemental oxygen, their O2 saturation, which should be around 95 to 98 in a healthy animal and a healthy human, drops to 60%. In a human hospital, if you drop there, it's considered a life-threatening emergency. So, keeping our patients warm, keeping them oxygenated, again, cascades through, because if you damage the cardiovascular respiratory center because of your anesthetic protocol, again, your data is compromised. It can take hours to days to even weeks for that damage to recover in an animal if we're using a poor anesthetic protocol. And again, the whole point of the study is to get good data. Now you compromise your animal. So I know I've jumped around a little bit. Any one of these pieces would be something that you could probably spend a few hours on. The resources are out there. So my goal has been to provide you a whole bunch of things to just start considering, because any one of these you can follow down. And again, this is from CompMed, the ALAS web service, tremendously. There are textbooks out there, especially for lab animal surgery, just a general primer. Mike Swindle's works on pigs is fantastic. Even though it's on swine, the basic principles and the reasons that he's using them translate across the species. But if you're working with surgeons, make sure they're doing things right. Because if they're not, broken record, I admit it, but your data is compromised. And it's a cliche, but it also happens to be true in the lab animal surgical field. How I do my work will affect the study. And after a while, ethically, we do this, but the saying is, you tell me what results you want, and I can do the surgery in such a way to maximize the chance that we get them. We should be doing it properly. If you're a surgeon or an anesthetist, again, we have an ethical obligation to our lab animals. We have an ethical obligation to the humans that our drugs and devices will go into. We have to give our best efforts. We have to refine our techniques, because the ultimate goal is to gain the highest quality data from every animal. Every animal that's going to be sacrificed has to be done humanely. We have to keep in mind and focus on proper welfare and
animal, we also have to make sure that the sacrifice that they're going to make is for good data. Because otherwise, it's a waste of their lives, it's a waste of our time. And again, if you're on a discovery team, it can be a waste of years of your work. With that, that's all I have to say, or at least all I have time to say it in. So thank you very much, and please send your questions, and I will answer them to the best of my ability.
Thank you, Matthew, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through May 27th, 2019. And as a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Bye.